It talks about all the physical activities that's happening inside the heart based on the electrical impulses that stimulate the contraction and relaxation of the muscles of the heart itself, right? Then of course we already talked about the blood vessels, which act as the conduits for blood to be delivered to all the cells and tissues of the body, okay? Now, this portion here is referring to the overall physiology of the cardiac tissue itself, of the heart and all of that stuff. So therefore, the cardiac output is defined as the amount of blood that is ejected out of the heart per minute time, okay? So it's measured in mils per minute. It's a rate type of activity, right? So typically, a normal cardiac output is going to be roughly about 5.5 liters of blood per minute. Okay, if you have to convert this to milliliter, that's 5,500 milliliter per minute. That's a lot of blood to pump out in a minute time. And that's under normal resting condition. When you're engaged in exercise, this can increase up to 5 times that amount, okay? All right, so now the cardiac output itself is regulated by 2 factors, and these 2 factors directly change the cardiac output. One of these factors is what we call the heart rate, and the other one is called the stroke volume, right? We already talked about the stroke volume to some extent, because the stroke volume is the amount of blood that is ejected out of the heart per heartbeat, per cardiac cycle. So every single time the heart relaxes and contracts, it pumps out some amount of blood, and we call that the stroke volume, and that's the reason why the stroke volume is measured as mils per beat, okay? How much volume of blood per heartbeat is ejected out. The heart rate is measured as beats per minute, and we know to regulate the heart rate, this is all due to the activities of your SA node. Okay, because the SA node is your natural pacemaker, basically the electrical activities that is produced by the SA node will regulate the electrical activities throughout the whole heart. So therefore, if the SA node produces 75 action potentials per minute, then your heart rate is 75 beats per minute. Because every single action potential essentially stimulates all the conduction system to basically match that electrical activity, okay? So, there are ways for you to increase and decrease the heart rate. There's ways for you to increase and decrease the stroke volume so that you can maintain this number in this amount of rate-dependent volume all the time. Okay, let's say you're under some kind of resting condition, whether you're a normal person or you're an extreme athlete or whatever the case is, right? You always have to try to maintain this amount of volume being pumped out the heart every minute time. If this volume decreases, then you're gonna have some particular condition that sets in, because if you don't eject out 5.5 liters of blood per minute, then you may not have adequate delivery of oxygen and nutrients to every single cell and tissues of the body. So therefore, certain organs and stuff like that will start to suffer, i.e. your kidneys, your liver, your lungs, whatever the case is, right, will start to suffer. And if it suffers for a long period of time, then you get into medical conditions that we call some kind of pathology. Okay, so you have kidney failure, liver failure, things like that, right? So you have to maintain this blood flow, this high rate of flow all the time. Okay, so how do we regulate the heart rate? How do we control the SA node? What do you guys think? The SA node is just a bunch of autorhythmic cells, right? They are intrinsic cells. That means they have the ability to spontaneously produce action potentials. So you leave them alone, they can actually produce action potentials. But there's also some kind of external influence that will regulate the intrinsic activity of these autorhythmic cells. What do you guys think that is? The what?
The vagus nerve.
Okay, the vagus nerve, which is part of what? Which is part of what system?
The parasympathetic.
Parasympathetic, which is part of your motor activities, right? Now, in addition to the vagus nerve, what else can regulate the heart rate?
Exercise.
Because the vagus nerve is what? Parasympathetic? What opposes parasympathetic? The sympathetic. So if you want to increase the heart rate, then you apply the sympathetic activity. Okay, so if there's some sympathetic activity that's applied to the SA node, then this will increase the amount of action potentials produced per minute time. How do you change that action potential? So you guys remember, let's say if this is coming from the SA node, right? The action potential looks like this. Phase 4, 0, 3. Which phase can you change by applying the sympathetic nerve? That's the only phase you can change. Okay, so if you apply the sympathetic nerve, this slope changes to this. So it reaches threshold much faster. If it reaches threshold faster, you can produce more action potentials in the same amount of time, okay? So instead of your resting heart rate of, let's say, 75 beats per minute, now you can apply the sympathetic activities to increase that to 120 beats per minute. Does that make sense? This is the reason why when you exercise, there's an application of your sympathetic activities to your SA node so you can increase the action potential being generated by your SA node. And that's the reason why when you exercise, you have an increase in heart rate, okay? Or when you have some kind of fear, anxiety, you also have an increase in heart rate because the sympathetic activities is also being applied to your SA node. All right, now if you want to decrease the heart rate, then you apply your parasympathetic activity. If I tell you the intrinsic activity of the SA node, so let's say if the SA node left alone produces up to 115 action potentials per minute. Okay. What can you apply to the SA node to bring this down to a normal level? Because a normal resting heart rate is about 75 beats per minute. So what can you apply to this SA node so that it can now produce 75 action potentials per minute instead of 115? Because if your resting heart rate is 115 beats per minute, that is sinus tachycardia, and that is abnormal, okay? So therefore, it has to be anywhere between 100 to 60 beats per minute, within that range, to be considered to be normal. So how would you bring the SA nodal activity down to 75 action potentials per minute? What do you apply? What do you stimulate the SA node with? Parasympathetic. All right. So let's say here you have parasympathetic activities, right? Applying its activities to the SA node, bringing it down. Which parasympathetic activities or which nerve is part of this parasympathetic pathway? It's called the vagus nerve. So therefore, the vagus nerve is very important as it acts on the SA node to bring the heart rate down to about 75 beats per minute. This is the resting heart rate. Okay, this is not like you're trying to slow the heart rate down. This is just basically bringing the activities of the SA node down to a normal physiological level. So therefore, the resting heart rate is maintained by, first and foremost, the overall activities of the SA node and the vagus nerve, reducing that activity down to about 75 action potential per minute. So in terms of the resting heart rate, we call this under the effect of the vagal tone. Okay? Which basically means the vagus nerve is responsible for the maintenance of this resting heart rate. So therefore, if you apply the vagus nerve, what is it gonna look like? Which phase can you change? So you're gonna do this. Right? So therefore, parasympathetic increases the duration of phase 4, which is your pacemaker potential, Therefore, the amount of action potentials that is produced in the same amount of time is now decreased overall. Less action potential is being produced, therefore you can actually bring this SA node of activities from 115 action potentials per minute down to about 75 action potentials per minute. Does that make sense? Okay, that's very important. If you have little children, go home and check the heart rate of your children. It is much higher than 100. Right? You ever listen to heart rate of little kids? Like my kid right now is just going, like massively fast, right? So that's because they don't have enough of this vagal tone yet. Eventually, they will develop this vagal tone activity. The parasympathetic nerve will have a higher action to it, and therefore bring the SA nodal activities down to 75 action potential. Now, here's another question for you guys. What neurotransmitters are being secreted here by the vagus nerve to stimulate that same node? Are you sure?
Acetylcholine.
Are you sure?
For parasympathetic?
I don't know. You tell me. Which one do you think it is? It is parasympathetic, right? So what's released by the postganglionic neuron?
Norepinephrine.
Are you sure?
Yeah.
Who agrees with Anton?
Thanks.
I'm just asking. It has to be a consensus, right? All right. You think it's acetylcholine?
For parasympathetic.
Okay. Are you sure? Who agrees? Who thinks it's acetylcholine? Good. It's acetylcholine. Now, the acetylcholine that's released by the postganglionic neuron binds to what receptor on the SA node? Are you sure? I mean, ligand gated is the nicotinic receptor. What's the other one? What's the other receptor specific for ACH or for acetylcholine? Besides nicotinic. Muscarinic. Good. So which one do you think it is? It's only one, not both. Why do you think it's muscarinic? Because what is the activities of nicotinic receptors? You guys, you better know this. Because I will ask you this question. Because you've already learned this stuff last time. And I've already tested you guys on this. So every single internal organ now is dependent on the activities of sympathetic and parasympathetic input. So therefore, you still have to know all of the stuff that is involved with the autonomic nervous system, right? All right, so if it's acetylcholine, you said it binds to muscarinic receptors, right? Why? Why can't it bind to nicotinic receptor? Yeah, because nicotinic receptors are always excitatory. In this particular instance, you're trying to bring the activities of the SA node from 115 action potentials down to 75 action potentials, which means you have to actually inhibit the activity of the SA node. And the only receptor that can bind to acetylcholine that can do that is muscarinic receptors. Okay? So in this particular case, the muscarinic receptor will bind to it. What neurotransmitters released between the pre and the postganglionic neurons here? Acetylcholine. Now, what receptors are on these postganglionic cells that binds to that acetylcholine within this autonomic ganglia? Cholinergic just basically means that it binds to acetylcholine. That would be the nicotinic receptors, okay? Because you always want to have excitatory activities between these two. What is the name of that ganglia? No, the ganglia itself. What's the name of the ganglia where the pre and the post ganglion neuron synapses for the vagus nerve? Terminal ganglia. Why is it called terminal ganglia? Because it's located on the SA node. It's located in the heart itself. And that's why it's called the terminal ganglia. Okay, good. That's your quick review. Please know it. All right. Sympathetic activity increase the heart rate. Parasympathetic activities decrease the heart rate. That's simple enough, right? Now, if you increase the heart rate, and you wanna keep this cardiac output the same, what can you do with the stroke volume? So this is the math that you need to do. There's no numbers involved. I mean, the only numbers really is this thing, right? There's no actual numbers that you can plug in. But based on this equation, I can give you different conditions, and then you have to figure out what's gonna happen to the other factor, what changes you need to implement in these factors to either maintain the cardiac output the same or increase or decrease the cardiac output appropriately. So therefore, let's just say in this scenario, you want to keep your cardiac output at 5.5 liters per minute, but for some reason your heart rate has gone up. What can you do to keep this number the same? Decrease the stroke volume. So how can you change the stroke volume? Well, there are 3 factors that directly influence the stroke volume. Let me circle these 3 first. So for sympathetic activities and parasympathetic activities, it has direct influence onto the heart rate because it directly stimulates the SA node to do what it needs to do. Okay, so for the stroke volume, the first factor that can change the stroke volume, change how much blood you can actually pump out of the heart, is called the end diastolic volume. So in the heart, it depends on, for this factor, it depends on how much blood you fill the chambers of the heart. Because the more blood you fill the ventricles, the more the ventricles will stretch. And when the ventricles stretch, it also stretches the sarcomere of those cardiac muscle cells. When those sarcomeres stretches and you stimulate them, they will contract back with greater force. Okay? You guys remember that particular theory that we talked about in skeletal muscle? It's called the length-tension relationship in skeletal muscle. Length tension, basically the length is the length of the sarcomere, and depends on how far the sarcomeres are spread apart. If you stimulate it, it will contract back to produce a tension, right? So in skeletal muscle activity, the optimal length of a sarcomere is the resting length of the sarcomere. In cardiac muscle activities, the actual optimal length of the sarcomere is actually beyond the resting length of cardiac muscle sarcomere. So therefore, in the length-tension relationship in cardiac muscle, we don't call it length versus tension, but the length of the sarcomere is dependent upon how much volume of blood we fill the left ventricle. So we call this left ventricular end diastolic volume. This is the amount of blood that we fill the left ventricle at the end of ventricular relaxation. Now, when this left ventricle contracts, it's going to eject volume. And the amount of volume that is ejected out of the heart is dependent upon how strong that contraction is. So therefore, this is now known as the stroke volume. Okay? In cardiac muscle activities, if you increase the amount of blood that fills the left ventricle, you will increase the stroke volume until it decreases the stroke volume. Okay? We call this the Frank-Starling curve. It's named after Dr. Frank and Dr. Starling, who stole this from their graduate students. If you want to make, receive good data, hire good graduate students. Right? If you guys ever go to a UC system in California, the UC system, when they make their exams to weed out 95% of the students so that they can select the top 5% as graduate students for their research. Right? And that's why most of the exams in the UC system is based on a bell curve shape, which means that top 5%, that's going to be recruited. The vast majority of the class will get C's, right? The low 5% will get an F, but the top 5%, that's the ones they're looking for. So when I went to UC system, I hated the exams. And I remember in one of my organic chem class, I got a 32 out of 100, and I still got a C. Happiest person in that class. It doesn't matter, right? particular professor, I mean, he was already like higher up, he doesn't know what he's doing. But he made his test so difficult, it was 4 questions, and it's worth 100 points. That means in organic chem, you start with some kind of substrate, you have to do all those steps. If you miss one of those steps, you're screwed, because you're not gonna get the yield that you want, like the percentage product that you need. So therefore, you're gonna lose all that point. So he's trying to see who has the ability to cognitively make, all these products for him. That's how he recruits his grad students. Anyway, that's just a little side note. It's not part of the test, by the way. So. All right, so for this particular Frank-Starling curve, right, the more blood you put into the heart, the more you stretch out the sarcomere. So for this, let's say if the left ventricle is completely collapsed, right? Let's say you have very little volume in it. If you have little volume of blood filling the left ventricle, then when it contracts, it will not eject out a high volume of blood. Make sense? If you continue to stretch this or allow for the ventricles to relax and fill it with blood, let's say if you fill it enough with blood to reach its resting length of the sarcomere of those cardiac muscles, and you stimulate it, it will contract to give you this amount of stroke volume. Okay? So this represents the resting length of cardiac sarcomere. In skeletal muscle, that was the optimal length. That basically means in skeletal muscle, you need to relax your muscle completely before you contract it again to produce the highest amount of tension. In cardiac muscle, you can actually stretch that sarcomere a little bit more by putting more blood into the left ventricle. So essentially, once you put more and more blood into the left ventricle, you reach a stage somewhere around here where it can produce the highest amount of stroke volume for you. So this is called the optimal length. So the optimal length for cardiac sarcomere is actually beyond its resting length. Under normal physiological conditions, even if you're exercising, you will always reach that point and no more. You will not go beyond that point. But when you have pathological conditions setting in, like let's say if you have some kind of damaged cardiac tissue, and the tissue starts to scar over, and basically how the heart replaces itself is like to put more sarcomeres in a lengthwise condition, so therefore it causes the muscle wall, the myocardium, to thin out, and it dilates out the chamber of the heart. So if that's the case, then you can actually fill more blood into the left ventricle. But because once you go beyond that point, you have lost some degree of muscle. So therefore, your stroke volume, the amount of volume you eject out of the heart, will actually decrease. Okay. So as far as we're concerned, the end diastolic volume if you increase the end diastolic volume, you will always increase the stroke volume under normal physiological conditions. We're not talking about pathological condition, right? This is when we have left ventricular dysfunction, things like that, like heart failure, okay? But under normal physiological conditions, according to the Frank-Starling curve, when you increase the end diastolic volume, you will always increase the stroke volume to a certain point, okay? Now, how do you increase the end diastolic volume? Yeah, you return blood to the heart. So what do we call that? What do you call the process where blood returns back to the heart?
Venous return.
The venous return. So if you increase the venous return, you will also increase the end diastolic volume. If you increase the end diastolic volume, you will increase the stroke volume. If you increase the stroke volume, you will increase the cardiac output. Make sense? Okay, if heart rate is left alone. How do you increase the venous return? Well, there are 6 factors that will increase or decrease the venous return, or will influence the venous return to a certain extent. So the first factor is the pressure difference. So pressure difference between falling asleep back there. Between the arteries and the veins. Okay? So between the arteries and veins, there's a pressure difference that will drive the blood continually from the arteries to the veins, therefore continually return the blood back to the heart. Okay? The second factor is that there's a pressure difference between the venules, which are the smallest veins, and the largest vein called the vena cava. Okay. There's a difference of about 10 millimeters mercury between these two type of veins. So therefore, blood continually moves from the smallest venules into the vena cava, which is right next to the blood. So therefore, if you increase this difference, you increase the venous return, you increase the end-diastolic volume, you increase the stroke volume, you increase the cardiac output. Okay. The third factor is if you apply the sympathetic activities. So when you apply the sympathetic activities, you can stimulate the smooth muscles that's wrapping around the veins. Even though there's not a lot of smooth muscle, you can still stimulate the smooth muscle, right? So when you stimulate the smooth muscle around the veins, it induces what we call venoconstriction. Instead of vasoconstriction. Vasoconstriction refers to constricting the arteries. Venoconstriction refers to the constriction of the veins. Okay, so if you apply the sympathetic activities, you will increase that venoconstriction. Basically, the veins kind of clamp down. That pushes blood back to the heart. Does that make sense? Okay. A fourth factor is something we already talked about, right? So we have a combination between the skeletal muscle pump working in conjunction with the venous valves. You guys remember that? Right? Skeletal muscle pump, basically your skeletal muscle, the veins are between your skeletal muscle, so when you contract your skeletal muscle, it compresses on the veins. These valves allow for blood to move progressively back to the heart and prevent blood from moving backward into the capillaries. Prevents pooling of the blood so that blood can move toward the heart. So therefore, the harder you apply the skeletal muscle pumps and hopefully your venous valves are still intact, the more venous return you get. And if you increase the venous return, you increase the end-diastolic volume and so on and so forth. Okay? All right, the next one is ventilation or breathing. So we'll just call it breathing. We haven't gotten into the technical terms yet. So inhalation, increase the venous return. Exhalation, decrease the venous return. Okay, so therefore, if you want to do that, then you can increase or decrease the venous return. The sixth factor is to simply change the blood volume. The blood volume is the amount of blood that you have. How do you quickly increase the blood volume? What's that? Blood infusion, saline infusion, coconut juice infusion, whatever you want, right? Because coconut juice is actually isotonic. And like in extreme cases, let's say you're in the desert, you don't have saline, I don't know why you have a coconut, but let's say you do, right? You find a large enough needle, you can tap in that coconut, you can tap into your vein, lift that coconut above your heart and allow that fluid to drain. And you'll be fine. Okay? And it's a good amount of glucose in there to kind of also satisfy a lot of the glucose requirement that you need as well. So 6 factors that will affect the venous return. When you apply these 6 factors, do they affect the stroke volume?
Yes.
No, they don't. They only affect the venous return, which in turn affect the end-diastolic volume, which then affects the stroke volume. Application of these do not affect the stroke volume directly. Okay, so it's stepwise. Okay, now the end-diastolic volume, I forgot to mention, is also referred to as your preload. Essentially, this is how much volume you load into your heart. So the more volume you load into the heart, the more stretching it occurs, and the higher the contraction it produces. So it contracts harder then essentially it will pump out more blood. Does that make sense? So as you stretch the heart out, it's gonna contract harder back. Okay? All right. So end-diastolic volume, also known as your preload, can increase your stroke volume. The next factor that can affect the stroke volume is what we call the total peripheral resistance, which is your TPR. Okay? The total peripheral resistance, also known as your afterload, is essentially produced by all of the resistance in every single blood vessels that you have. So imagine if you have an individual, right? If you draw an individual, and the individual is like this. Okay? That's an individual. So imagine his heart right there. And the heart has an aorta right there, right? And all the blood vessels are distributed out. So you have a bunch of blood vessels going up to the head, blood vessels going down the arms, blood vessels coming down here, blood vessels dividing out into the legs, lower legs, and stuff, all that stuff. Okay. Now, if you take every single one of these blood vessels and you measure the resistance, you have a resistance here, resistance there, resistance there, 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 there. Okay, resistance there, resistance there. Okay. Did you ask why it's resistant? Anytime you walk, there's always a coefficient of friction that resists your movement. It's based on the structure. So therefore, if there's a blood vessel, it's actually a structure. So when blood is moving within the blood vessel, the walls of the blood vessels actually retard the movement of that blood. That is always the case under gravitational effect, right? That's physics. That's basically something that is always there. So inherently, anytime you move against any kind of object or any kind of surface, there's some kind of resistance to your movement. And that resistance is also due to the size of the blood vessel as well. So if the blood vessels dilate out, there's less resistance to the blood flow. If the blood vessels constrict, there's more resistance to that blood flow. Make sense? So therefore, all of these resistance can be added up together. Once you add all of these resistive forces together in every single part of your body, because all of these blood vessels are connected to your aorta, therefore, you can measure this pressure in the aorta as the total peripheral resistance. Because these are all the resistance in these blood vessels that is delivering blood to the periphery of your body.
So if you were to bend your arms and legs, that's gonna increase the resistance.
Not necessarily, because you can always dilate those blood vessels too. So the body's gonna try to find some nice happy medium for all of that activities, right? But if you have a bunch of, let's say, constriction that's going on, then you will raise that resistance very high. Make sense? So if that's the case, so let's say if you're doing that exercise and you're like constricting, your muscle, you're gonna increase the resistance. Not as much as you would do with the veins. Because the blood vessels, the arteries, actually already have a huge amount of smooth muscle that can actually regulate its own diameter, more so than the veins do, right? So we'll talk about this in the next chapter, in chapter 14, how we increase or decrease the resistance of blood flow throughout the blood vessels and stuff like that, because you guys have to do it too. But in this particular scenario, if you take all of these resistance and you add them all together, so the total peripheral resistance, in a sense, is equal to the resistance number 1 plus resistance number 2 plus resistance number 3 plus ... Right? In actuality, that's not the true equation, but let's just say that it is. So therefore, this total peripheral resistance is the pressure that is sitting in your aorta. So remember the cardiac cycle that I drew out? Why is the aortic pressure so high compared to all those changes in the heart? Well, because that is the pressure applied by the total peripheral resistance. What's the minimum total peripheral resistance under a normal situation? Roughly about 80 millimeters of mercury. When you guys take a blood pressure or assess a blood pressure, what's the diastolic number? Somewhere around 80 millimeters of mercury. Wonder why that is? Well, because your total peripheral resistance is based on the blood pressure. So it's systolic and diastolic blood pressure all the time, right? Because that's how much volume of blood is ejected into the blood vessels versus how much volume of blood is taken away from the blood vessels. Okay. We'll talk about that later too, because you have to assess the blood pressure at some point. All right, so this total peripheral resistance is known as the afterload, which means this is the amount of pressure that the left ventricle has to work against. So when the left ventricle contracts, it has to generate enough tension to overcome this afterload. So if this afterload is low, when the muscles contract, can it contract quickly or can it contract slowly to produce adequate amount of tension? If your load is low, how quickly can you contract to basically reach that load, reach the amount of tension to overcome it? Fast or slow?
Fast.
What do we call that relationship? Remember skeletal muscle physiology? What do we call this? So this is how quickly your sarcomere can shorten to produce tension to oppose a load. Okay? So this is called the force velocity curve. You guys remember that? Yes. Forgot a lot of that. That was like a long time ago. I was like last month. But you're right, it does seem like a long time ago. Okay, so in terms of this, think about this. Now let's say apply this to the heart, right? If you have a lower total peripheral resistance or afterload, as the left ventricle contracts, here's the load. So imagine here's the load, and you're trying to overcome this load. If this load is low, then the shortening, the time that it takes for the left ventricle to contract is really not that high, based on this curve. Right? So therefore, if the load is low, then the shortening velocity is actually high. So if the shortening velocity is high, how quickly can that ventricular pressure overcome the aortic pressure? Very quickly. So then that actually will shorten your phase 2 of your cardiac cycle, which is your isovolumetric contraction phase. Make sense? Because that phase is the phase that is required for the tension in the ventricle to build up enough so it can overcome the aortic pressure. And then that will allow for your semilunar valves to open to eject the blood into the aorta. But if the afterload is high, then it will take much longer for that left ventricle to contract. Because now as it contracts, it's still generating tension, but it's not enough, so it has to continue to contract. So it takes much longer, right, for it to contract. So the shortening velocity is very, very low because it's not longer. And as the ventricle eventually finally contract enough to reach or overcome the aortic pressure, too much time has passed during phase 2. So in this particular case, if you have an increase in afterload, your phase 2 is increased. When your phase 2 is increased, what happened to phase 3? It's significantly shortened. Okay, so if you draw out the cardiac cycle, one, two, three, four, five. Right, this one. Let me set the aortic pressure first. So let's say aortic pressure is like this. Okay, if that's the aortic pressure. And the ventricular pressure is here. vibration